So you've designed a brand new medicine, and in a test tube, it works perfectly. But what happens when you put it inside a living, breathing human body? That right there is the billion-dollar question. Today, we're diving into computational ADMT modeling, which is basically using computers to predict a drug's entire journey through the body before a single molecule is even synthesized. Take a look at this number. It represents a massive graveyard of failed medicines. See, historically, nearly half of all promising drug candidates ended up failing in late-stage clinical trials. And think about the cost. Not just billions of dollars in research, but years of lost hope for patients. The crazy part? They didn't fail because they were ineffective. They failed because the body just couldn't process them correctly. This was the massive wake-up call that completely changed drug discovery. All right, so here's our game plan. First, we'll visit that drug candidate graveyard to really get a sense of the problem. Then, we'll map out a drug's journey, check out the digital toolbox scientists are using, meet the body's very active gatekeepers, and finally, we'll look at what the future holds for this incredible predictive technology. For decades, drug discovery really only focused on one thing: how well a molecule could hit its target. But that tunnel vision led to this graveyard of brilliant drugs that just failed. They failed because of something called poor pharmacokinetics, a fancy term for how the body absorbs, distributes, metabolizes, and gets rid of a drug. Understanding this whole journey, what we call a drug's disposition, is absolutely the key to avoiding that same costly fate. This slide really shows that crucial turning point. That huge failure rate forced a total change in strategy back in the mid-90s. The industry realized it had to start testing for ADMET properties. That's A, absorption, D, distribution, M, metabolism, E, excretion, and T, toxicity, way earlier in the process. It all started with lab tests, or in vitro screening, and that generated all the data we needed for the powerful computer models or in silico models that we rely on today. So what exactly is this journey that we're trying to predict? Well, it's all captured in what's called the ADME profile. It's an acronym that describes the 4 major steps a drug takes inside the body. And let me tell you, getting this profile right is the difference between a blockbuster medicine and a complete clinical failure. This chart here really maps it all out. The journey begins with absorption, moves on to distribution all throughout the body, and then finally ends with excretion. For our discussion today, we're really going to focus on modeling this disposition pathway, predicting how a drug gets absorbed, distributed, and eventually removed. The journey always starts with the first big hurdle for any pill you swallow: solubility. It's a simple concept, but it is absolutely non-negotiable. If a drug can't dissolve in the fluid of your intestine, it has zero chance of being absorbed. It's kind of like trying to drink a rock. It's just not going to work. Okay, so once it's dissolved, the drug faces its next big challenge: permeability. It has to physically get across the wall of the intestine to make it into the bloodstream. And this isn't just some simple soaking through process. Sure, some of it is passive diffusion, but the intestinal wall is also lined with some pretty sophisticated machinery, active transporters, that can either help a drug get in or actively grab it and throw it right back out. So how in the world do we predict these super complex processes? Well, scientists have a digital toolbox with 2 main approaches. They're actually really different from each other, but both are essential if you want to get a complete picture of how a drug might behave. I absolutely love this analogy. On one side, you've got the statistical detective. This is the QSPR approach. It basically uses heavy-duty statistics on massive datasets to find patterns between a molecule's structure and its properties. It's fantastic at predicting what will happen, but it doesn't always explain the why. On the other side, you've got the master locksmith, which is pharmacophore modeling. This is a mechanism-based approach that tries to figure out the exact 3D shape and chemical features a drug needs to fit into a specific biological lock, which explains how it all works. Note, the data that feeds these models can be either 2D or 3D. Most models use the simpler 2D information, like a flat chemical diagram, because it's fast and pretty effective. But 3D models, which look at the molecule's actual shape in space, are often way more predictive. So what's the catch? Well, they need a ton more computing power, and that can be a real challenge when you're trying to screen millions of compounds at once. Okay, now let's talk about one of the most dynamic and challenging parts of drug disposition: active transporters. These are not just passive holes or channels. They are sophisticated molecular machines. They're the body's bouncers and gatekeepers, and they can completely change a drug's fate. So the really crucial thing to get here is that our cell membranes aren't just simple walls. They're actually patrolled by these gatekeepers. You have influx transporters that literally pull molecules into a cell, which can be great for absorption. But then you have efflux transporters that do the complete opposite. They find foreign molecules, like drugs, and pump them right back out, creating a major roadblock for that drug's effectiveness. And the most infamous of all these efflux transporters is P-glycoprotein, or PGP for short. This molecular pump is an absolute game changer. It sits in your intestine and literally ejects drugs, which limits their absorption. It lines the blood-brain barrier, preventing drugs from ever reaching the brain. And in cancer cells, it can cause multi-drug resistance by pumping chemotherapy drugs out as fast as they can get in. So you can see why modeling its behavior is so incredibly critical. And here, we can see how that master locksmith approach works in practice. This is a pharmacophore model for a PGP inhibitor. It's the blueprint for the key. Every feature you see mapped out here, like a water-faring region or a spot that accepts a hydrogen bond, is a critical piece of the puzzle. Scientists use models just like this one to predict if a new drug is likely to get ejected by PGP, and they even use them to design drugs that can block it. All right, so we have models for solubility, for permeability, and for individual transporters, but the real power comes when you put them all together. The future of this whole field is all about integration, creating these systemic platforms that can simulate a drug's entire journey. But as you can imagine, there's still some major challenges to overcome. This is how it all comes together. Modern software platforms use something called a CAT model, which stands for compartmental, absorption, and transit. Basically, they simulate the drug moving through the gut, from the stomach into the intestine, and in each section, they apply the models for solubility, permeability, and all those transporters. The end result is a much more complete, holistic prediction of how much of that drug actually makes it into the bloodstream. But here it is, the biggest hurdle the field is facing today. It's not a lack of clever algorithms or computing power. As the source material makes crystal clear, the weakest link is data quality. These computational models are only as good as the experimental data you train them on. If that data is inconsistent, or if there just isn't enough of it, the models won't be reliable. You really have to think of high-quality data as the fuel for this entire predictive engine. So, where's this all headed? Well, the field is pushing hard to model a much wider array of transporters, not just PGP, but others like BCRP and the OETP family. There's also a big move towards customizable models that companies can train on their own private chemical libraries. And really, the ultimate goal is the complete integration of all ADMET properties into a single, seamless system, with a much greater focus on understanding the deep mechanisms of why drugs behave the way they do. And that really brings us to the final thought. The ultimate goal here is pure prediction. The dream is to be able to take a drug design on a computer screen and, with a high degree of confidence, predict its entire pharmacokinetic journey. We're not there yet, but with every new model and every new high-quality data point we collect, we get one step closer to making drug discovery faster, cheaper, and a whole lot more successful.